Hello everyone, I'm Maria and I'm an ICASE PhD student at the Qualcomm Institute, and I'm here to give you a brief summary of my poster. So as most of you know, biocides are widely used to prevent and control hospital-acquired infections, but the emergence of biocide tolerance and cross-resistance to antibiotics is a major challenge. So for this project, we want to investigate the impact of prolonged disinfection exposure to two common nosocomial pathogens and to basically inform how to better control hospital infections. So for this, I did an in vitro based evolution model where the isolates, Staph aureus and Enterococcus faecalis were exposed to two commonly used biocides, chlorhexidine and octanidine, for around 250 generations. And what we saw was that these isolates were able to adapt increasing concentrations of chlorhexidine and octanidine even beyond the MIC points, and similar results were observed in both isolates. But at the end, the growth stopped, so they were very poor growers. So after seeing these changes over time, we wanted to phenotypically assess and study different traits such as biofilm formation, fitness measurements, and biocide antibiotic cross-resistance. So we phenotype all these evolved isolates, a total of 308 isolates from the different time points, and we saw differences in biofilm biomass. So for example, here you can see that when the Enterococcus faecalis mutants were exposed to chlorhexidine, they had lower biofilm formation after being exposed to this stress when compared to octanidine stress, and similar results were observed with Staph. And there was no major fitness deficit observed and no apparent cross-resistance to any antibiotics. So in general, octanidine show less potential to select for tolerance than chlorhexidine. But then we wanted to know the genetic basis behind this adaptation. So we selected samples from the late time points and we sequenced these samples. And what we actually identified in these mutants were SNPs repeatedly found in all these isolates and loss of function changes in Staph aureus and Enterococcus in a gene that is involved in phospholipid biosynthesis. And this was quite interesting because analogous changes were also observed in the same homologous gene in Enterococcus faecalis. So this strongly suggests a potential role of this gene in biocide tolerance. So now I'm in the process of characterizing the role of this novel gene and see what I can find. And so yeah, I'm gonna be on Monday at the Microbiology Society annual conference. So just come see my poster and ask me any questions.